one, and then we separate the sister chromatids from one another in meiosis 2. And again, the benefit of meiosis is that we're producing these genetically different offspring from one another, or at least we're generating the gametes that are genetically unique, so that when those genetically unique gametes come together, they make a genetically unique individual or offspring. All right, so here is again comparing mitosis and meiosis. So you're still going to have replication occurring in both. So you're still going to start with diploid organisms. In mitosis, again, the whole point of mitosis is just to separate those sister chromatids. And they're going to be genetically identical to one another because all we did was just make a copy of the DNA that was already there. And so the whole point of mitosis is to separate these sister chromatids, and after one cell division you end up with two cells that are both going to be diploid. Whereas in meiosis, again, you're still going to have replication, but you're going to have this crossing over occur within prophase 1. And then in the rest of meiosis 1, again, you're going to separate these homologous chromosomes from one another. And then in meiosis 2, this is where you're actually separating these sister chromatids from one another. So at the end of meiosis, you're going to end up with 4 genetically unique gametes, all that have one set of genetic information that are going to be haploid. All right, so there's something that's related to meiosis. And so when these homologous chromosomes are lining up in metaphase after crossing over has occurred, the way that they line up with respect to all of the other chromosomes is going to be totally independent and totally random. So in other words, there's different combinations of the way these homologous chromosomes can line up in metaphase. So if we're looking at a cell that has 4 chromosomes, such as in this example, so we have 4 different possible chromosomes. And so the way that those homologous chromosomes align can be completely different. So in this example, we have all of moms on one side and then all of dads on the other side. So when you separate it, you end up with 2 cells, one that has all of moms and one that has all of dads. Or what can happen is you can have this be totally randomized. So in other words, you might have one homologue from mom on this side and then a homologue from dad on this side. So in other words, it's the way that these homologous chromosomes line up with one another that's also going to result in genetic diversity. So you can see the difference between the gametes obtained in this possibility versus the gametes obtained in this possibility. And so every chromosome pair is going to orient itself just completely random and without respect to the other chromosomes. So this set of chromosomes doesn't care what these guys look like. It's only worried about itself. And so it'll just arrange randomly. It's just a total random process. And so again, that's introducing, or a different way to introduce genetic diversity into your gametes. And so there's actually a calculation that will allow you to calculate the various different combinations of possible offspring that you can get. And so whenever you look at, if we actually go back to this slide, right here, so you can use this formula right here to determine how many different possible gametes there are. And so the N is representing the number of sets of chromosomes. So we have one set of chromosomes here and one set of chromosomes here. So in humans, if you want to figure out how many different genetic possibilities there are, it would be 2 to the 23rd power. And that comes out to be roughly 8 million. So 8 million different genetic possibilities for each one of these gametes, because each set of chromosomes can align in only one of 2 ways. And so when you calculate the total possibilities of all of the arrangements of all 23 pairs of chromosomes, you end up with 8 million different genetic possibilities. So each time we undergo meiosis to produce that sperm or that egg, there's 1 in 8 million chances of getting a particular genotype. So this is why we all look so different, yet we're, similar in the same ways. And so tons of genetic diversity being introduced in meiosis. And another way to introduce genetic diversity is through what's called random fertilization. So if each one of our gametes is unique for 8 million different ways, or in 8 million different, gametes, I suppose, then that means each sperm and each egg is going to be 1 in 8 million, right? And so depending on which sperm fertilizes which egg, if each sperm is going to be different and each egg is going to be different, depending on which ones match up during fertilization, that's also going to dictate what the genetics of the offspring is going to be. And so if each gamete represents, this is that 8 million number that we came up with, this is 2 to the 23rd. So if each gamete represents 8 million different chromosome combinations, then at fertilization, so this is 8 million for the sperm and 8 million for the egg, that means during random fertilization, there's over 70 trillion different genetic combinations that can be made during fertilization. And that's because your egg is going to be unique compared to 7,900,000, whatever this number is minus 1. And so when you multiply those together, this is the odds of a particular egg coming together and the odds of a particular sperm. And so again, you're having 70 trillion different options of genetics in your possible offspring. And then again, this crossing over is going to increase this genetic variation as well, because you're switching different chunks of DNA between mom's chromosome and between dad's chromosomes. And one thing in this process, in addition to being called crossing over, it's called genetic recombination. So if we look at this example right here, all right, at the end of meiosis 2, we end up with 4 gametes. 2 of them are going to look just like the original parents did. So that would be this guy and this guy. And then the other 2 guys, these are going to be the ones that swapped over pieces of their DNA. And so we call these guys recombinants. They're recombinants because their DNA was recombined. And so at the end of meiosis, you're always going to end up with 2 parental phenotypes and 2 recombinant phenotypes. Do y'all know what phenotype means? A phenotype, so there's genotype and phenotype. The genotype is literally what your genes say. Like if I have blue eyes, then my genotype for that gene would be little b, which we'll get into. But so that's my genotype. My phenotype is what it actually shows. So my phenotype would be the blue eyes, but the genotype is dictating the phenotype. And so these guys are going to look just like the parents because they didn't change anything. They didn't, move any of their DNA around. But these recombinants, because they have different pieces of DNA associated with them, these are going to be those recombinants. And so after each round of meiosis, you're going to end up with 2 parental strands that look exactly like the parents, and then you're going to have 2 that are going to be different from the parents. And those are going to be these recombinants. And it's those recombinants, again, that are going to help introduce this genetic variation into the cells. Okay, so again, in meiosis 1, we have homologous chromosomes lining up. They're going to cross over in meiosis 1, and then they're going to be separated from one another during anaphase. And then in metaphase 2, again, now you have these sister chromatids that are lined up, and they're going to be separated. And so you can see the two recombinant pieces of DNA right here, and these are the parentals right here. Questions? Okay. Really, again? Okay. All right. Comparing mitosis and meiosis. Mitosis, one cell division, starting off as diploid, ending up as diploid. No genetic diversity whatsoever. All we're doing is we're making a clone of a pre-existing cell. Whereas in meiosis, we're making the sperm and the egg, and we're introducing that genetic diversity in prophase 1 of meiosis 1, which is that crossing over and making these recombinants. And then again, in meiosis 1, we're dividing the homologous chromosomes from one another, and then meiosis 2, we're separating the sister chromatids from one another. Oh, okay, that's it. Questions?